This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 52 for September 30th, 2009. I'm your host, Vincent Racaniello, and today I'm talking with Dr. Scott Hammer. Dr. Hammer is chief of the Division of Infectious Diseases and the Harold C. New Professor of Medicine, also Professor of Epidemiology at Columbia University Medical Center. Dr. Hammer earned his B.A. in 1968 from Columbia University and completed medical school in 1972 here at the College of Physicians and Surgeons. Most of Dr. Hammer's career has been devoted to improving the treatment of HIV infection. He has been an active investigator in the Adult AIDS Clinical Trials Group, sponsored by the National Institutes of Health. In the 1990s, he chaired the two largest antiretroviral trials of this group, which helped advance combination therapy as the standard of care worldwide. Dr. Hammer is also an investigator in the NIH-sponsored HIV Vaccine Trials Network, a multi-center organization whose mission is to develop an effective preventive HIV vaccine. He's the protocol chair for the Partnership for AIDS Vaccine Evaluation 100, also called PAVE, a Phase 2b clinical trial to evaluate the efficacy, safety, and immunogenicity of a multi-clade HIV-1 DNA plasmid vaccine, followed by a multi-clade recombinant adenoviral vector vaccine in HIV uninfected persons. He currently is a member of the Executive Committee of the AACTG and chair of the Phase 1-2 Committee of the HIV Vaccine Trials Network. He has served as an advisor to the Swiss HIV cohort study, the French Agence Nationale de Recherche sur le Sida, and the HIV Netherlands, Austria, Thailand Research Collaborative. He's vice chair of the IAS USA Antiretroviral Guidelines Panel and editor-in-chief of the World Health Organization's Antiretroviral Guidelines for Resource-Limited Settings. He also serves as a member of the Governing Council of the International AIDS Society. When the results of the HIV clinical trial in Thailand were recently released, and which we discussed on TWiV episode 51, I asked Dr. Hammer if he would speak with me about AIDS vaccines and how they are tested by clinical trials. And I think you'll agree that Dr. Hammer is the right person to talk to about this. I'm very happy that he agreed, and I believe our conversation will be of great interest to listeners of TWiV. Please stay tuned after our conversation for a few brief announcements. I'm talking today with Dr. Scott Hammer, who's chief of the Division of Infectious Diseases at Columbia University Medical Center. Thanks for talking with me, Scott.
My pleasure.
I didn't realize you were a purebred Columbia person.
Yes.
You went to Columbia undergrad and medical school here as well.
Yes, I did, although I did broaden my perspective a little by doing 1 year of residency at Stanford and infectious disease training up in Boston.
So California didn't seduce you away from New York?
It would have, but I was already committed to coming back the year after the senior residency.
Are you a New Yorker?
Yes, originally.
Wow. Well, you're about my generation, right?
Yes.
I came here in 1982, and you had already come back here.
82, no, I was still in Boston. I had just, actually when I started my faculty career at the Deaconess Hospital after fellowship.
So was that your first experience with HIV and AIDS in Boston?
Yes, I was. I finished my fellowship. I had trained with Marty Hirsch at MGH and was doing herpes simplex work. Then took a job with a new infectious disease division that was being put together at the Deaconess Hospital, the New England Deaconess Hospital at the time, now the Bethersville Deaconess Medical Center, one of the Harvard teaching hospitals, one of the smaller ones. And they were starting a division of infectious diseases, and it was my first faculty job. And as we got established with the first AIDS cases were getting admitted to the hospital, and I gradually, like several other individuals, moved from herpes simplex cell interactions to looking at HIV in the laboratory first, in vitro interactions with cells, antiviral therapies in vitro, cytokine issues, and then gradually got involved in clinical trials and moved into the clinical trials of therapeutic agents with the AIDS Clinical Trials Group, and then the rest is history.
Yeah, so you have been working since then on AIDS.
Yes, it's been amazing, actually, and a privilege in one sense, to see a disease come upon us of such magnitude and instilling such fear and stigma and everything about this disease, which affects every part of human existence and world security, etc. That point with a new virus, and then the etiologic agent being described, antiviral agents being developed, being able to test them in people, see them approved, and then watch the disease turn into, for those in the developed world, a chronic manageable illness where it was a death sentence in the first several years of existence.
It's really unprecedented to start with a brand new virus of such magnitude, of such importance, and to go through all this, and it's been since the early 1980s now. So what I wanted to focus with you today on is vaccination and clinical trials, because as you know, there was a big result in Thailand last week announced, and a lot of our listeners are curious, and since my understanding is limited, I thought I would come to somebody who's been doing this since the beginning. And it has been since, I understand that vaccine trials have been ongoing since the 80s. So my first question is, we have great vaccines for many other virus infections. Why can't we get one for HIV? What is the problem?
So you'll get many answers to that question, but one area where I think you will get consensus among all people involved in this, from basic scientists to advocacy groups, is the diversity of the virus and its ability to escape pressures, selective pressures, whether it be our own adaptive immunity, cytotoxic T-cell escape, or escape from neutralizing antibodies that we may produce, or drugs that we may give. And the diversity of the virus, which is obviously most expressed in the envelope gene, the envelope protein of the virus, which is where you would want to develop an immunogen or a vaccine candidate to develop neutralizing antibodies, if you will, or a cytotoxic T-cell response. The escape it's difficult. And probably the most important thing of that is what we've got. We haven't developed, as we have for other pathogens, an immunogen that will create broadly reactive neutralizing antibodies. So if you take the viral diversity most expressed in the envelope of the virus, and then our ability to develop a mimic of the external glycoprotein that binds to the cell and develop neutralizing antibodies that are broadly reactive across the 9 major subtypes of HIV, we haven't been able to do that. And that's really one of the holy grails of vaccine discovery and development now. And so what the field has been focused on is, quote, T-cell based vaccines that, at least in non-human primates, induce a CD8 and CD4 cell responses, and we can go into that in more detail, but primarily what people are looking for are strong CD4 help and solid CD8 cell responses to limit virus expression. And in non-human primate models, so far, for the most part, these T-cell based vaccines do not protect rhesus macaques from infection, but modulate the level of viremia post-infection, maintain CD4 counts, and delay disease progression.
Is it necessary to have a vaccine which would protect against all the subtypes? In some countries, there are predominantly one or a few, correct? So if you start from the assumption that maybe in some areas we could use just a single subtype, is it possible to have an immunogen that induces protection against just one?
So that is an interesting question and a big debate in the field. Some people think, yes, you can have subtype-specific viruses, but subtype-specific vaccines. But even within a particular subtype, there's ongoing diversity that's going on. And although people have looked at vaccines, for example, the Merck vaccine that was tested in the STEP trial, which we can talk about, a phase 2b study that whose results were announced 2 years ago, where the vaccine didn't work and actually may have caused an excess of infections, or at least been associated with an excess number of infections. That was a subtype B virus vaccine, and was being tested also besides the United States and the West. It was being tested in South Africa, predominantly Clade C area. So there are cross-reactive immune responses that you can see, but whether they're protective or not is not clear. The other issue that goes on here is that there's a continuing mix of, it's not just subtype diversity, but in melting pots of the world, particularly Africa, there are recombinant forms being developed, seen all the time, where two subtypes can infect the same individual, infect the same cell, and through a couple of cycles of replication, create a mosaic that, if then gets transmitted, can establish a new recombinant form that may have elements of both subtypes throughout its genome. And in particular, in the envelope, would actually be throughout the genome. So I think as people think about vaccine development, the concept of cross-subtype immune responses hopefully will work, because we can't cover all the different subtypes and their variants. But whether one subtype will work or whether a few subtypes will work to cover the waterfront is a debate. Now, there are many interesting approaches to this. One approach to look for a single vaccine type, if you will, is called the common ancestor. And Betty Korber and others, Betty Korber from Los Alamos and others have tried to look for, through phylogenetic analysis, and Jim Mullins also from Seattle, try to come back to a virus that might be termed the final common ancestor from which everything derived. And would that virus engender enough cross-reactivity to work? It's an interesting concept. I think if you look at the phylogenetics variation that we've seen in HIV, that's would be nice, but I think there's lots of questions about that. And I think ultimately people think we'll need a mixture of clear subtypes to create a broad immune response that'll, it may not be exactly the virus in the vaccine, but close enough to develop a protective immune response.
I'd like to interrupt our conversation to acknowledge the support of this episode by Citrix, the makers of GoToMyPC. Now, GoToMyPC is what we call a remote access software package. It allows you to access a computer when you're not in the same room using the internet. If you're, for example, a frequent traveler for business or pleasure, you know that you still need to keep your business running, which means you need reliable access to your home or work computer. And the easy and secure way to do that is GoToMyPC, brought to you by Citrix. It's a very easy solution. On the computer that you want to reach from anywhere, you go to Citrix website, you install a small program, and then you obtain a login and a password. And this is completely easy and totally secure. And then from any internet-connected computer, you go to their website, to Citrix's website, enter your login information, and you'll see the desktop of your computer where you installed the original software. You can access all your files, you can run programs, and even collect your email and access networked resources. Now, this sounds like something that you would love for your business or pleasure. You can try GoToMyPC free for 30 days. Just visit gotomypc.com/podcast. That's gotomypc.com/podcast. You'll get a free 30-day trial. And of course, we thank Citrix for their support of TWiV. So in the Merck trial that you just talked about, what was the immunogen used?
So the immunogen didn't contain any envelope. It was internal structural or regulatory proteins of the virus. It was GAG, POL, and NEF from a subtype B delivered by a recombinant adenovirus 5 vector. It was highly immunogenic in adenovirus 5 seronegative individuals, creating 85% T-cell ELISpot responses, for example, one of the hallmarks of immunogenicity that the field had used in humans. And it had worked to a reasonable extent in non-human primates, but we can get back to what role non-human primates have as far as predictive models. But it didn't work when tested both across the population in about 3,000 individuals. And what it showed was it was actually stopped formally by a Data and Safety Monitoring Board for futility because it wasn't going to make its endpoints. And its endpoints were two. One, would it prevent infection, a classic vaccine protection, and the second was whether it would modulate the viral load after infection in those who became infected. And it was clear from a Data and Safety Monitoring Board in the fall or so of 2007 that it was not going to be successful. And the trial, so vaccinations stopped, and the trial goes on in follow-up, but the vaccination stopped. And what was troubling about the result was, as far as there was no difference in viral load between vaccine and placebo recipients, but there was a higher number of infections in the vaccine recipients than those who received placebo. When you look at the subgroups and do various statistical modeling, the major factor that seems to be associated with an increased risk of acquisition in the vaccine group was not being circumcised. Mm-hmm. But also what early on looked like an association, and was an association, although not clearly statistically significant, was the pre-existence of immunity to adenovirus type 5. And the highest risk of being infected in a vaccine recipient was being both uncircumcised and being Ad5 seropositive. Mm-hmm. Mm-hmm. And that created all sorts of issues, because what was that, a true association? What was going on to make this, to increase the risk of acquisition? And we still don't know the answer to that. There's a lot of data on circumcision and in men and with randomized trials that it protects men from heterosexual acquisition in Africa with 3 randomized trials. But the preexistence of immunity to the vector was previously only thought to be a problem in neutralizing the vector and maybe making the vaccine less effective. And nobody really thought that it would increase the risk of infection. And it's not clear that it really did. We're still looking for this. There are a couple of theories that came out when this result appeared. One was that preexistent immunity to the vector caused a memory response, which activated CD4 cells, which then may have been homed to the mucosal epithelium, and then you were having activated CD4 cells in the target area of where the virus would be able to infect them easily with sexual exposure. And people have tried to mimic that in animal models so far, and I haven't seen any clear results. And as the study has gone on, over time, the Ad5 effect is waning. And whether the pre-existing immunity to Ad5 was real, it certainly was real early, it's less real now. Mm-hmm. But whether it's a marker for something else or a true etiologic risk factor is not clear. But it's sort of shaken up the field.
Was that the first T-cell vaccine trial, or have there been others?
That was the first T-cell based vaccine trial that came to a public result, because already the Thai trial was enrolling.
I see.
At the time.
The Thai trial didn't use adenovirus. It used canary pox and a purified protein component.
Yes.
So which of those would have been analogous to the Merck immunogen? or neither?
So let me take a step back for a moment. So before the Merck result came out, there were 2 previous trials of GP120, the envelope glycoprotein, just as using repeated injections of GP120,
Right.
and those were the VaxGen trials, and both of those failed, basically showed no protection.
And those were aimed at inducing antibodies,
Yes.
correct? So before, I'm sorry to interrupt, but I want to just clarify one thing. When you do these trials, do you measure antibodies in the recipients? Do you check them if they're neutralizing in cell culture? And in that case, there are no neutralizing antibodies generated? Or is not as simple as
so
that?
the standard of monitoring in whether it's a early phase trial or a late phase trial, but particularly late phase trials, when we're looking for markers and of immune response that we may relate to activity, is we'll look at the T cell responses by typically ELISpot or flow cytometric analysis for intracellular cytokine staining. Those are the standards right now. And on the humoral side, we look for antibody. We typically look for binding antibody to an antigen on an ELISA plate or to neutralizing efficacy in vitro. And products like GP120, the VaxGen products that were tested, would develop you can develop antibodies, neutralizing antibodies to just, more in animals than in people, to one strain, but it's not a broadly reactive, cross-reactive neutralizing antibody. What people do develop are pretty uniformly, 95 to 100%, substantial and high titers of binding antibodies, which previously had been thought not to matter, or who knew what they were really doing. But that question now has come back with the TIE trial. So to get back to your question about what was being tested in the TIE trial, it's an interesting combination. So it's part of an approach to vaccines, for HIV vaccines, of a prime and a boost. And we've looked at, over the course of years, in non-human primate models, different primes and different boosts. And sometimes they're homologous and sometimes they're heterologous, either in how the antigen is delivered or what you're delivering. Are you delivering a viral vector with genes, or are you delivering the protein? And what genes have you selected? What protein subunits have you selected as well? And so, for example, the Merck study did a prime boost with the same recombinant adenovirus 5 vector with the GAC-POLNF genes in it from subtype B. The TIE trial did a prime boost with a viral vector first, a canarypox vector, Alvac 1521 from the Sanofi Aventis, or Sanofi Pasteur rather, and that vector contains HIV GAG genes for coding GAG, and POL from subtype B, and an envelope from a subtype E, or to be more specific, what's circulating in Southeast Asia and in Thailand is an AE recombinant, which has been termed as subtype E, but it's really an AE recombinant. And then the boost in this regimen was a protein boost of GP120, a binary GP120 that had a subtype B GP120 and a subtype E GP120. And it was a total of 4 injections of the canarypox vaccine over 6 months, and the last 2 injections of the canarypox were combined with an injection of the GP120, the binary GP120, the VaxGen product. And when this study got started, it was actually very controversial because the Alvac vector, the canarypox vector, had been only modestly immunogenic by our classic techniques in human studies, with approximately 30% ELISpot activity, and if I remember correctly, and we already knew that the VaxGen product by itself had failed in the large-scale phase 3 trials. So why would they work together? And there wasn't clear-cut definitive non-human primate data. The trial went forward. It was controversial at the time, but the result now is very interesting because it didn't show a difference in viral load post-infection in those who got infected, but did show a 31% reduction in acquisition. Mm-hmm. Now, this trial enrolled 16,000 individuals in Thailand in general communities in 2 provinces, I think Rayong and Chonburi in Thailand, which were men and women of general risk. These were not specifically targeting high-risk populations. The risk varies across the population, but overall it's just a general risk population in a country that was developing a generalized epidemic, but it really has been under control, so it's nowhere near the prevalence of South Africa, for example. And they've been able, with a number of issues, to lower the prevalence throughout the country. 100% condom use and constant counseling, etc., of the population. So of 16,000 individuals, if you look at the total study, less than 1% became infected. And so they had 125, if I recall correctly, endpoints, acquisition endpoints, and there were 74 infections in the placebo recipients, mm-hmm. and 51 in the vaccine recipients. And so that's a 23-person difference and this has been debated as what this means, but is a statistically significant difference and a 31% reduction. The absolute difference, you can argue, is modest, but the relative difference is a statistically significant result and has reshaken the field. And I think the team is working very hard to develop liaisons with advisors from around the world, really, as to what to look at next. They have immunologic results, which they'll be presenting at the AIDS vaccine meeting in Paris later in October, when the trial is formally presented. And that's all we've seen so far are preliminary results, essentially, the basic results of the study. But the details of the study will be presented, I think, on October 20th in Paris, and those will be very interesting to look at.
I presume it'll be published at some point eventually.
Yes, hopefully the team is looking to try to submit and hopefully publish for peer and have it appear in the peer-reviewed literature soon. But even with whatever they've done so far, the result was unexpected, and it will take time to go back to the specimens that have been stored and decide what other experiments need to be done and what subpopulations may have been protected within this group to figure out what happened.
Is the number of individuals who got infected 1%? You said is that
just under 1%?
Is that unusually low for a trial of this size?
It's interesting that when they first started the study, they predicted a higher incidence, but then as it diminished, which is a good thing, of course, in Thailand over time. And that made that they had to expand the sample size to actually, because in order to get the number of endpoints to make the trial non-futile. So typically, if you go for an efficacy trial and to get answers in a reasonable period of time, you'll go into populations of higher risk or in a generalized high prevalence, high incidence area where you can get the answer in a shorter period of time. But this was a number of factors that led to this Thai trial, which has a long history of cooperation between the United States and the Thai military and the Ministry of Public Health in Thailand, with other disease entities and longstanding cooperation. And the push of the Thai government and the Thai people, really, to be part of clinical trials and solutions and looking and being very pro-vaccine development. They tested many vaccines in Thailand quite successfully. One of the things about the result, which we'll have to look at, is whether, as I alluded to earlier, there's a subgroup that was protected. And there are ways in which one can think about this. Are these people who may have been exposed to a lower virus inoculum,
Mm-hmm.
which of course retrospectively will be impossible to determine. But what will be possible to determine is whether there's an HLA link.
Right.
And for example, even in the trial of the Merck vaccine that failed, the STEP trial, which we talked about, there are subpopulations that have protective HLA haplotypes that the numbers are small, but look like in those individuals, the vaccine in Ad5 negative individuals may have actually modulated viremia. So even within a trial that overall was a result that shook the field negatively, there are hints that we may have something to go after, including, for example, the fact that early on in some of the infections in the STEP trial, they've been able to show through viral sequencing that there was some selective pressure on the virus by the vaccine response, which is an important. So some pressure was put on the virus, but not enough. And those similar studies will be done now with the TWiV trial. One of the issues is there aren't a huge number of specimens, I think, as far as cellular materials stored, is my understanding. And so they know they have to use the specimens wisely to try to answer this question.
To look at HLA type, you have to have cellular specimens.
Yeah, well, yes. I mean, HLA you can do over, with, you need a cell, but you don't need a lot of cells, obviously, to do HLA typing. If you want to look at new tests of cellular immunity, including a new assay that is being bandied about as something potentially important, in vitro virus inhibition assay, you need a lot of cells. So they've got my understanding is they've done ELISpot results, and they have other cellular immune results, which they'll present at the AIDS vaccine conference. But if we want to go back now and look at other new cellular immune assays, the issue is like with any study, were enough cells put away to do the assays, and can they be done on frozen cells? And the team very wisely has, as I mentioned earlier, asked many people from all areas of basic virology and immunology through clinical trialists for suggestions as to how to prioritize what the questions are in this study, recognizing how important it is. And this leads to an issue that is also a very important term in vaccine discovery and development, and that is there a correlate of protection? And that's what we're talking about. If you have a protective response by a HIV vaccine or any vaccine, but let's say you can protect people from infection or you can modulate their viremia post-infection, is there a marker of that effect that you can measure? And once you get that and you prove it in an efficacy trial, then you can use that marker as its own endpoint in future vaccine development. Does another vaccine develop this response in people? One of the negative surprises of the STEP trial was that we had put our hopes on intracellular cytokine and ELISpot, and as I mentioned already, good responses were engendered, but they weren't protective. And when case controls were done to look at infected versus uninfected people, they couldn't tell the difference in those who develop immune responses. So it doesn't look like ELISpots, at least for that vaccine, are a candidate for a correlate. So what is the correlate? We don't know. And if probably the longest lasting effect of the TWiV trial will be if a correlate of protection can come out of this, then it will really advance the field. There's another overriding result of this trial, which I admit to bias about because I'm a clinical trialist. But after the STEP study, there was an appropriate resettling of the field, and things sort of halted in the clinical trials arena for a while. And appropriately, there was more emphasis on going back to basic concepts and trying to figure out what is it about this virus and the immune response that we don't understand. as well as trying to strengthen the effort to develop a neutralizing immunogen. Most people think we'll need both T-cell responses and B-cell responses to control this virus, but clearly to prevent transmission, we will need a neutralizing immunogen. And that shift, quote, back to basics, as it's called often in medical science, was important and appropriate, but there was almost a denial that clinical trials help us, and that we should only do large-scale clinical trials if we've got a product on a strict development to licensure path. And I think what the STEP trial showed us in a negative way, and what the TWiV trial is showing us in a positive way, is that predictions are often incorrect. And the non-human primate model has been good, but it's not 100% by any means predictive of human immune responses. And what we have to do is be smart about what products we take into advanced phase 2 testing, so-called 2B trials, with efficacy or activity endpoints. But that's a real part of vaccine discovery. And I think that has given a boost to the field that we need a balanced approach. And of course, there are issues of resources and cost, and it obviously costs a lot to do a clinical trial, but we're only going to get there if we select wisely and continue to do clinical trials that will tell us in humans whether we've got something, however promising it looks in non-human primates.
One of my listeners, we get a lot of feedback on TWiV, and one of them wrote, is it possible that some of the infections were caused by different serotypes in this trial, that the effect might have even been better if you remove that? And is that something that they could tell from the samples?
It's so the virus side of the equation is crucial, and they're going to, for sure, look at all the breakthrough virus isolates in those who became infected and sequence them, probably do full genomic sequencing for two things. One is, as per the question, to look at the epidemiology of infection in the individuals who got infected, how closely related within the subtypes that circulate in Thailand, which are subtype B and the AE recombinant, as well as to look at whether there's any pressure on the virus, even in the vaccine breakthroughs. Those two things will be very important. Also, phylogenetically, if there's a cluster of infections that are epidemiologically related, which we have no hint about yet, that can also relate to the interpretation of the results. So the sequencing of the virus isolates in those who became infected is a crucial part of the epidemiology of the trial and whether the vaccine was doing something.
Right. Now, you said earlier that the result reshook the field. But a lot of people, of course, look at these numbers and think they're so close that they're not demonstrating any efficacy. So why is it such a landmark in the vaccine, AIDS vaccine field?
So I'm biased on this because I'm a clinical trialist, but I think the field was so down and suppressed after the Merck results that we needed more balance. And I think that the investigators, to their credit, if you look at their public statements and their press releases, have called this a modest effect. They have not said this is a grand slam. This is clearly a 31% effect. If it's statistically significant, is not what we look for. We like to see 80, 90% efficacy of vaccines. But this is a vaccine combination that was thought to produce a zero effect. And you still have to explain the result. Even though the numbers, the absolute difference is small, if you look through the medical literature, this is not uncommon that low prevalence events, you do large numbers of studies, cardiology trials and other trials, where you may have to enroll lots of people to get a study done in a relatively doable time period, 3-5 years, and the number of events are relatively small actual clinical endpoints, and what is the significance is in the relative difference. So this is no different than many other types of trials, and this was a very well done study with good randomization, superb follow-up, I think over 90% follow-up to the end of the study, and the analysis was an intent to treat analysis, modified intent to treat, which meant you had to receive at least one of the vaccinations and not be infected at baseline. That analysis is a very pure type of analysis, and unless you come up with an explanation for why there's this difference, its chance is possible, but it's actually a 1 in 40 chance you would come up with this. This gets into statistical issues, but it was a two-sided test of significance that was used for this vaccine study, which means they were looking for either efficacy on acquisition or enhancement. And if you just you would do a one-sided test if you were just looking for efficacy and not worried about enhancement, but they appropriately did a two-sided test, which for the non-statisticians just means in simple terms that it's actually a more robust result than statistically than it appears just by the p-value, which is 0.039. And I think it's a little bit, you could do it in a number of trials to say, well, if 5 people moved from the one group to the other, it would change the result. That's true of many studies. And I think it's not the case here where this vaccine is going to immediately leap to licensure. No one is thinking that's going to be the case. We're even not sure that this vaccine combo will be tested in another confirmatory trial. What it does tell us is that we have this result, which no one expected, and to ignore it would be really a wrong approach. And what we should do is take a step back and not just dismiss the results, but decide how we can effectively confirm that this is real, and to take that information and to use better immunogens and more improved tests to try to really come up with a next better vaccine. And again, the way to look for this will be deeper analyses of the data and going into subpopulations, doing the host genetics, doing the viral sequencing, using the samples that are stored for appropriate testing to try to come up with either a correlate or an explanation that confirms this result. And then there are also some thoughts, which is appropriate, to use the non-human primate model in a circular fashion. So typically, vaccine candidates are tested in vaccine and non-human primate models and then go into humans. But there's been a lot of talk in the field about getting the non-human primate scientists to work more closely with the clinical trialists and to actually have a feedback loop. And there are already discussions going on about how this regimen can be adapted to the SIV macaque model and be tested more thoroughly in macaques to try to see if we can reproduce this result and understand it in an animal model. Because we're not about to the field isn't about to open up another 16,000 person study. So I don't think we should ignore the results, whether it's a landmark. We'll have to see. My own cautious optimism is to say it's a potential landmark. And yes, I think it's boosted the field, even though there's skepticism and appropriate skepticism from some quarters. But there's difference between skepticism and dismissal. You can be skeptical of the results, as scientists should be, until they're confirmed, but open to the fact that preconceived ideas may be incorrect. And so dismissal is something we shouldn't do, and we should take this result and run with it as far as trying to confirm that there's something here. Ultimately, I admit there may not be anything here, and this may not be a landmark. But we've now had 2 trials that, one, we predicted success and never predicted it would make infections worse or as far as numbers are concerned, and it did, and totally surprised the field in a negative way. Although I would mention that even that study, the secondary analyses that have come out of that study, have been extremely important. And if we hadn't done that trial in humans, we would still be whistling in the dark. And no one wants to see any harm in any trial, or excess infection, numbers of infections in a clinical trial of a vaccine candidate. But the volunteers in that trial have contributed substantially to vaccine discovery and development, because we would be happily marching along the same path if we hadn't had that result and the field didn't take a good look at itself and retool. By the same token, this trial, as I mentioned earlier, gives us a positive result, which very few people predicted. And that just means that there's unpredictability. And again, I would say with resources and other judgments to bear, as we develop new vaccine candidates, we have to decide what are our criteria for putting them into advanced 2B testing. Now, 2B testing doesn't have to be 16,000 people. If you go into higher risk populations, you can look for efficacy with smaller numbers. And there is a trial ongoing now that is looking at a different prime boost regimen that was developed by the Vaccine Research Center of the NIH, which is a DNA prime recombinant adenovirus 5 vector boost regimen, which is being tested in the HIV Vaccine Trials Network, of which I'm an investigator, and the study is open now, and actually we're recruiting here at Columbia for this study. And it's gone through quite a history of development, because the study was going to be a 12-country, 8,500-person study, and was scheduled to open in September 2007, and never opened because 10 days prior to its opening date, the step trial results were announced. And through revision and careful analysis of the step results, and thinking about whether it's worthwhile to proceed to an activity trial, is what we're calling it, eventually came to pass 2 years later, essentially, that we are testing the vaccine only in the United States in men who have sex with men, or MSM population, in the Vaccine Trials Network. So it's a high-risk group, and it's 1,350 individuals. Now, the study is powered actually to look at modulation of viremia. Mm-hmm. We will look for acquisition differences, but we don't have great power to answer that question. But that's a vaccine that is, quote, multi-clade, multi-gene. So the DNA prime has GAG, POL, and NEF from subtype B, and envelopes from A, B, and C, the major circulating subtypes in the world. And the adenovirus 5 vector boost is GAG and POL fusion, codes for a fusion protein of GAG-POL, doesn't have NEF in the boost, but does have envelopes from A, B, and C in the adenovirus 5 vector boost. Now, this is a different adenovirus vector than was used in the Merck trial. It's got the Merck vector had a deletion in E1. This vector has a deletion in E1 and E4 and a partial deletion of E3. So it produces fewer adenovirus-specific proteins, therefore may be less immunogenic in individuals as far as inducing adenovirus immunity to the vector, if that's a problem. And we don't know whether that is a problem. But what we've done is take a very safe population derived from the STEP trial. So we're only enrolling circumcised men who are seronegative for adenovirus type 5. So no pre-existing immunity to adenovirus type 5. And if you look at the STEP trial, that population had absolutely no increased risk of infection in the vaccine group. If anything, the relative risk was 0.7. I had a confidence interval that crossed 1, but there was clearly no increased risk. So we're doing this trial, and it's now open in sites across the country. And I think we've gotten a boost, if you will, a shot in the arm, to use the vernacular, and vaccinology from the TWiV results, because it just helps show that the importance of testing something with an endpoint that then you can relate to activity or efficacy. Now, I would mention that this trial called the HBTN 505 trial is not on a path to licensure, again, because now it has the adenovirus vector in it, and people are wary of that after the STEP results. So this is not a licensure development trial. This is a scientific experiment that is for vaccine discovery and trying to determine whether there is in vivo activity of this vaccine regimen, and to do it in a way that will help the field and advance the field. Well, I think it, in the same manner in which TWiV stepped up to the plate for the RV144 TWiV trial, people in the United States want to be part of the solution and think beyond themselves when they enter clinical research trials. I will say, as an investigator and a clinical researcher, it's actually quite impressive, whether it's a phase 1 trial or a phase 2 or a phase 3 trial, to ask people who are seronegative for HIV to enter a trial and get immunized. Now, there's no way you can get HIV from these vaccines because they're just pieces of HIV, but still, these are healthy people stepping up to the plate to help the field. And in one sense, particularly, those in phase 1 trials are accepting the risk of something that's never been in humans before, possibly. In advanced phase testing, at least we have the safety profile of this. It's been in hundreds of individuals so far of this regimen, but people are still willing to roll up their sleeves and come in for many study visits, get a lot of blood drawn for the greater good of humanity, and it's very impressive.
I think that's a good point to make here, that these individuals who participate in these trials are really the heroes. They're tough things to assemble, but the people that you enroll are crucial.
It is, and so I've done a lot of trials in therapeutics, and the volunteers for therapeutic studies are, and certainly throughout the development of potent antiretroviral therapy, true heroes of the epidemic. But it's also a somewhat different issue as far as equipoise, in the sense of how an investigator or a trial clinician approaches things. In the early part of the AIDS epidemic, when people were dying of this disease, they were sick. We had drugs that were not approved yet. They had preclinical safety, and part of the randomization was to treat people. And you were treating people with disease in the hopes that you would find a better treatment. You had still very devoted to and always devoted to our participants, and gratified by the development of drugs. And now we have 25 approved agents against HIV in 7 classes or subclasses. And even in people with multi-drug resistance, we can suppress their virus to undetectable levels. And we wouldn't have gotten anywhere without volunteers stepping up to the plate.
Sure.
But there's still a contrast. When I moved into the vaccine arena, there's still a contrast to enrolling, particularly, as I mentioned, phase 1 studies, which we started here. People just saying, I want to do this because I have a friend or a relative who's gotten HIV or AIDS, or I just want to do it for the greater good. And whether we enroll phase 1, 2, or 3 trials, the relationship we develop with the participants is very close. And I remember your listeners may be interested in one anecdote, which was true at our site and true across the country. So we participated in the Merck STEP trial. And there was a very emotional moment when the results were announced, and we had to talk to our participants. And it even there was a lot of emotionality, whether it's our research nursing group, study coordinator, the physicians, scientists. And we were buoyed by the participants who said on more than one occasion, we knew what we were getting into. This is science and research. If I got placebo, I want to enter your next trial. And that's really impressive and is what carries us forward. So I think one of the larger take-home messages, particularly for maybe your younger listeners who are thinking about medicine or science as a career, is that clinical trial results, there's a lot that goes on in a clinical trial. And the interpersonal relationships that develop are, at least in infectious disease studies or things like HIV and AIDS, create bonds that are really quite deep and respectful. And there's more than just a paper and a result. There's lots of camaraderie and other things that go on. Plus, I would just mention we do very intense risk reduction counseling. We often find diseases in people or problems that when we do screening for studies and people who are not connected to the medical care system, that we may discover something, that we get them connected to medical care. If they should get infected during a trial, we make sure they have the best care available early on in their infection. So there's a lot of commitment to people. It's not just numbers. And I think that's an important point to make, and perhaps a discussion like this where you may have people thinking about what type of career they want to go into, as well as what is clinical research. And it's an interesting and challenging area.
None of this, of course, comes out from reading the numbers in the newspaper at all. I have no appreciation, nor even from reading the paper when it comes out. So that's great. And we do have people who think about careers in research and clinical science, so that's good to hear. Just, can you give us an example? How many, aside from the people enrolled in these trials, how many people such as yourselves and nurses and pharmacists, how many other people are needed for a trial of that size?
For example, the trial that we're currently doing, the 1,350-person trial, I can just tell you we have 14 sites across the country. And typically, it's a very multidisciplinary affair. So you'll have a principal investigator who leads the trial, often a sub-investigator. On the physician side, a nurse practitioner or physician assistant or research nurse, usually 2 or 3. A whole very important staff of community educators and recruiters to go out in the community and try to connect with individuals. You need laboratory technicians, you need pharmacists, and you need regulatory individuals who will interact with the IRB and make sure we're reporting things properly and all the data, and then a data manager. So it's usually a team of anywhere from 6 to 15 people, depending upon the size of the site and how many people are being enrolled.
So that's per site?
That's per site. So every site has a sort of basic requirement depending upon how many people are going to enroll in more than one trial, perhaps. But it requires all these disciplines to do it right. So it's a real team event. Nobody can do it alone, and the only way you can do excellent, high-quality clinical research is to have all these elements together. So investigators, research, nursing, study coordination, recruiters, educators, pharmacists, laboratory technicians, and regulatory experts. Otherwise, you can't do it.
So if someone wanted to do this for a career, wanted to be Scott Hammer and one day do clinical trials, what advice would you give them? So if they're early on in their educational steps.
That's a good question. I think I wouldn't advise anybody to be Scott Hammer. I think I would take derivatives of that because every career is unique, and clinical trials are a challenge. But a couple of points, and this is the beginning of this talk. At this discussion, I mentioned the uniqueness of having had a career that started essentially at the faculty level with the onset of the clinical AIDS epidemic, being a virologist, a laboratory virologist to start, and still a clinical virologist, and being very interested in the virology and the pathogenesis of this disease, moving into the therapeutic phase, and being interested in drug resistance, watching this whole thing evolve over time, and then moving into international events and trying to roll out treatment or help the WHO give guidelines to developing countries to roll out the benefits we see in the United States and Europe and Australia to resource-constrained areas, and then moving into the prevention field and vaccine. And I did that more for the clinical trial challenge of study design, and then got hooked on it because I'm not an immunologist. I'm a card-carrying clinical virologist. And I thought what was interesting about vaccine trials is that there were aspects of design that were an interesting challenge, as well as the fact that we were doing T-cell-based vaccine studies that weren't going to prevent infection, so people were going to get infected, and therefore being a treater on the physician side and a developer helping to do trials in treatment areas could be useful. Now with the potential and learning a lot from my immunology colleagues about the possibility of protection, it's really been a privilege. But nobody can predict that and I was in the right place at the right time and had wonderful mentors, and specifically Marty Hirsch at the Mass General Hospital has to be mentioned as first and foremost in this, because if you look at so-called leaders in HIV AIDS across the country, a very substantial proportion of them were trained by Marty Hirsch, whether that's David Ho at the Aaron Diamond AIDS Research Center, Bruce Walker. I could go on. The list is incredibly long. And so advice to people thinking about a career is first, listen to your heart and your head, in the sense that you need to be practical about what options are out there, but you also have to follow where your interests are. And I think I would say exploration is the most important thing. Before you make a decision, use your training to explore possibilities. So the route, for example, there are many routes to this, but the common route now probably is people who are interested as MDs or MD PhDs who want to do infectious disease fellowships and be infectious disease specialists. That's a route to go. And then during their fellowship training, to think about whether it's the more basic science or translational science or more pure clinical trials that they want to do, and whether it's domestic or international. And it's not easy to become a clinical trialist in the sense that for career development, because what are the opportunities? You have to think about where you are on the spectrum. Do you want to be part of a larger team? Do you want to do early phase trials? Do you want to do investigator-initiated studies? It takes a long time to generate results in clinical research, and the demands of academia may be difficult as far as accomplishments and individual landmarks that will get you promoted and get you a grant. But so I would say the way things are going these days is that there's a team and interdisciplinary approach. So many people, I think, are interested in HIV/AIDS at the clinical trial or clinical epidemiology level. They do infectious disease fellowships. They may go do epidemiology training at schools of public health. And they're really committed to both prevention and treatment and have an international perspective. So things are very different now. And over the last several years, what we've seen is we're training a whole generation of brilliant and committed individuals who are training in infectious disease and epidemiology and are linking to fundamental science and translational science and going out and working internationally, working with colleagues in developing countries where also young individuals who are being trained and funded by NIH and other mechanisms to develop in-country capacity, which is obviously where the epidemic is and where the solutions have to come so that we really eradicate this epidemic if we ever can. And that then requires it's a long answer, but that then requires a team approach, that you have to be part of a team and then think about what niche you have within that team. So what lots of our fellows do is they may be part of a larger group and understand about multi-center and multidisciplinary trials, but then they'll be doing a part of that there is their own area. They'll do a sub-project or a sub-study that will be something that they can put their effort into and be recognized and put the protocol together, link to the laboratory science, and analyze the results and develop a name for themselves or a reputation, and then build on that. So it's a long process, but it's rewarding. But you really have to have the desire to do it, and what you need is a good mentorship who can really help you decide what your talents are and where the openings are in the field. It's like everything else. It's a little bit of chance and luck as to where you end up and where you wind up and who influences you. And then I would also say that fellowship training is an opportunity also which we need to continue the fundamental science development. The whole issue, if we're ever going to get a vaccine, it's going to start in the labs of people like Dennis Burton and others, George Shaw, David Ho, variety of individuals who will make the discoveries at the basic level that will give us the neutralizing immunogen that we can take. And I would say that clinical trials, it's another comment, what's been so gratifying about it is being trained by someone like Marty in viral disease and viral pathogenesis and wanting to understand the science of the virus and its interactions, and work with a lot of people who try to answer the questions, not just does this drug work or does this vaccine work, but why does it work and how does it work and how does the body escape from its effect. And that constant thirst for knowledge, because we're all students if we stay in academics, is really the driving force. It's hard to plan. I think you have to sort of be flexible and expose yourself and put yourself in a position where you've got good mentors and flexibility of the program, because you may discover that your thoughts about what you wanted to do may change radically under the influence of a laboratory experience or an international experience.
You mentioned chance a few times, and it makes me remember Louis Pasteur, who said, chance favors the prepared mind. Dick Despommier is always bringing that up. It's always good to be well prepared, get a good education, and then when you're ready, come see Scott Hammer and train with him and learn how to be a trialist. Scott, I think our listeners will really enjoy this. I really appreciate you taking the time to talk to me, because your office is stacked with papers, and I know you're a busy guy.
Well, I thank you very much for the opportunity to speak to your audience, to speak to you. It's been a pleasure.
I want to thank Dr. Scott Hammer for taking the time to be on TWiV. At times the discussion was very technical, but as always, if you have any questions, don't hesitate to send them to us. You can send us an email, twiv@twiv.tv. You can also send us an MP3 file, or you can call up Skype, and the username is TWiV Podcast, all one word, and leave a voicemail. And of course, if you have stories that you'd like us to discuss, you can tag them in delicious.com with 4:TWiV Podcast, all one word, and that ends up sending those tagged stories to us. Now, as you might have gathered from our conversation, Dr. Hammer is very interested in training the next generation of clinical trialists. And in fact, he's program director of the Infectious Diseases Fellowship Program here at Columbia University Medical Center. And if you have any questions about a career in clinical trialing, I'm sure Dr. Hammer would be happy to respond. If you're a new listener to TWiV, please consider subscribing in iTunes. It's free and allows you to automatically receive each new episode as it's posted. If you don't use iTunes, you can always download episodes at TWiV.tv, where we also have extensive show notes. If you like TWiV, please tell others about it. Links and reviews in iTunes are particularly helpful. TWiV is part of sciencepodcasters.org and promednetwork.com, websites where you can find high-quality science podcasts from a wide variety of fields. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.